let's talk about how viruses modulate cell translation. We've talked about the cell's feeble attempt, I would say, to shut down translation to get rid of the virus infection. But as I've shown you, most viruses that are around today have gotten around that. Now let's look at how the virus regulates translation. And the goal here, of course, is to have the whole translational apparatus for themselves. And we're going to look at regulation of EIF4F, which again is a complex of EIF4G, EIF4E, and EIF4A, these three proteins here. And basically what we're going to look at is cleavage of EIF4G. So it's really one of the subunits of EIF4F. And this is done by viral proteinases, 2A or L. Now, this is done by viruses that have an iris. They don't have caps, so they don't need intact EIF4G. So let's see how this works. Here's an example in cells infected by poliovirus. If you monitor the rate of protein synthesis at different hours post-infection, in polio-infected cells, you can see the rate of synthesis declines very quickly by 2 hours, and then it's replaced by a new round of translation, which is viral protein synthesis. What happens here is that the host cell translation is shut off. You can see this on the gel to the right. This is a protein gel showing labeled proteins at different times post-infection with polio. You can see at 0 times there's a big smear of cellular proteins that are made. And by 3 hours, you can see that is starting to decrease in intensity as the virus shuts off translation. And then by 5 hours, you have mainly viral proteins made. So this makes perfect sense to humans anyway, that you'd want to shut off the host to get all of the apparatus to yourself. So how does this work? 2 virus proteases, either 2A in polio or the L protease in other viruses, cleaves EIF4G near its N-terminus. And as you can see, that takes away the EIF4E binding site on EIF4G. So capped messages can no longer be translated because you can't get the ribosome to bind, right? If you cleave 4G here, even if 4E could bind the capped message, it couldn't recruit the ribosome because all the 4G in the cell is now cleaved and it no longer has a 4G binding site. So that effectively shuts down cap-dependent translation. But it's brilliant, of course, because these viruses that do this have an iris in their genome. They're not capped, so they don't need this 5' EIF4E, or I should say N-terminal EIF4E binding site. It's very neat. Now, there are serious consequences of translational inhibition that go beyond what we have talked about so far. So let's say we have a cell which is making proteins. It gets infected by a virus. Now, so here's our circular polysome. It's got ribosomes translating a circular mRNA. A virus infects the cell. The cell responds by phosphorylating EIF2, or the virus cleaves, say, 4G, and this inhibits translation. So now we have what we call a stalled translation initiation complex, because these mechanisms of initiation mainly act at the level of initiation. These are sensed by the cell. The cell says, we have stalled translation complexes. And what it wants to do is sequester them into what are called stress granules. And it does so by making a number of proteins, or having a number of proteins. They're shown here in yellow, and these form structural components of the stress granule. So these are actually very discrete areas of the cytoplasm. They're quite small. You can see them in a light microscope, though, that contain these proteins and stalled translation complexes. And the cell's idea is that we're going to protect these RNAs until the virus is over or some other time. Now, of course, the virus does not want to have its mRNAs put into these stress granules, because these are basically silenced areas where no translation happens. So many viruses encode antagonists of stress granules. You can see all these viruses listed here antagonize stress granule formation. And for some of them, we know which proteins are antagonized specifically. So, for example, polio cleaves this particular component, G3BP, of the stress granule to prevent their formation. So viruses have evolved to antagonize stress granule formation. Now, when translation is inhibited in a cell, there's actually another pathway that the stalled complexes can take. So it can either go to stress granule formation. There are also another set of bodies in the cell called P bodies, which are similar. They contain stalled translation complexes. And again, viruses don't want to be put into them, so they antagonize their assembly as well. This pathway is accompanied by destruction, or can be accompanied by destruction of the mRNAs. So there are a variety of cell proteins shown in blue here that decap the RNA, take off the poly-A, and even degrade it. And this can happen outside or within the P-body. And again, these P-bodies are bad for viral mRNAs, so viral proteins have evolved to antagonize their formation.